Hi everyone, you've made it. It's the end of the semester. There's just a few more ends to tie up, and one of them is the final exam. So this is Professor Foster Scharf here to give you some tips and some hints on what to study and how to prepare. When you sit down to take your final exam, number one, please be certain that you set aside enough time to complete it. It should take you no more than 2 hours if you've studied properly and you're familiar with the concepts that are on the exam. So you want to shoot for 2 hours. Next, you want to make sure you take the exam in an environment that will allow you to focus. So if you have kids, you might want to wait until the kids are in bed, try to find a quiet place, and really just focus on this. Please make sure that you restart your computer before taking the exam, and when you open your web browser to actually start, please leave only the exam window open. I've had a couple of people during the semester have problems with their computer cutting off on them while they're in the middle of exams, and I don't want that to happen to you during the final. So take a few minutes, restart your computer, only leave the exam window open. Finally, make sure you have a stable internet connection, because once you start the exam and its components, you won't be able to reopen it if the exam winds up closing on you. Your final exam is due by May 13th at midnight. Please hand in your exam by this deadline. I'll need to submit final grades to the college within about 72 hours of this date, and submitting your exam on time gives me enough time to go through your multiple choice and your essay questions carefully. So the final exam has two parts. First, you have a multiple choice section. It's about 41 questions, and they're coming from a pool. So you've been working with these types of questions for the entire semester. Some of them may look familiar to you. You'll have about an hour and 15 minutes to complete this part of the exam. So there's more than 1 minute per question. Second part of the exam is essay. There are 3 essay questions, and they are multi-part questions. This part of the exam is not timed. However, when you sit down to take an essay question, you must complete that question in that sitting. So what I've done is I've broken up the essays into 3 different mini exams, so you could take these different essay questions at different times. You don't have to sit and take them all at once. Okay? Now, neither the multiple choice nor the essay are open book or open notes. They are purely open mind. I think the only exception to that is that in my lectures, I might have mentioned that you could use a 3x5 card, like a little cheat sheet, to help you remember the different antibiotics and their targets, as well as the antiseptics and disinfectants and their targets. For your 41 multiple choice questions, about 40% of it is microbiology basics. These are things that we've talked about in previous chapters. You've already seen them on exams 1, 2, and 3. 60% is only on this last part of the class, the antiseptics and disinfectants, your antimicrobial drugs, and the innate immunity. And for the innate immunity, the questions are coming from the lecture notes. So now I'm going to talk a little bit about things you should do to prepare for the multiple choice part of the exam so that this doesn't happen to you. Okay, so if I was preparing for the multiple choice section of this exam, I would go back, hint, hint, and look at a lot of the basic concepts. You know, those really basic things that someone should know if they've had a microbiology class. For example, at the end of a microbiology class, you should be able to give me characteristics for bacteria, fungi, protozoa, viruses, animal parasites, and algae. Those things were back in the first chapter that we did. So I would make sure I knew that. You might also go back and review the names and examples of these different groups of microorganisms. I'd also look at prions, by the way. You could get a question on that. I would go back and review bacterial structures especially the structures of the different types of cell wall, gram-positive versus gram-negative, know what's in them, how are they different, why is one considered to be thick while the other one is thin, why does one make you more sick than the other, those types of things. I would make sure I was familiar with any unique structures in microorganisms. For example, all bacteria have peptidoglycan, right? And all fungi have, well, not all bacteria have peptidoglycan, but probably like 90%. So it's a basic characteristic, and it's unique to bacteria. They're the only ones who have that. Fungi have things like chitin and ergosterol that are kind of unique to them. Viruses have unique structures. They have those spike proteins that help them get into cells. So I would pay attention to those types of things. I'd go back to that chapter we did on epidemiology. Remember that? We talked about things like what it means when an illness is acute versus chronic, what inapparent infections are, opportunistic pathogens. We talked a bit about the normal flora. We talked a bit about the different phases of illness, like we talked about like the incubation period, the prodromal phase, period of convalescence, all those different things. I'd go back and look at that chapter. We also talked about disease transmission there. Could be important, hint, hint. So that's like 40% of the multiple choice. The other 60% is dealing with topics that are more recent. So you want to look at your chapters on the antimicrobials, aka the antibiotics. You want to look at the antiseptics and disinfectants again as well. I would go back and look at all of my antiseptics and disinfectants and learn what do they do. Okay, this might be on your cheat sheet. I think I allowed you to put the target of the chemical on like a little note card that you can use while you're doing the exam, so you might want to look at that. I might pay attention to what types of conditions can affect how well a disinfectant or an antiseptic works. Remember, things like organic matter inhibit the activity of these chemicals. Time of exposure is important and is related to their effectiveness. I would do the same things for things like autoclaving. We talked about incineration, so other non-chemical methods of controlling the growth of microbes. You need to know what the targets are. Autoclaving, remember, it targets the proteins in the cell. It causes them to denature and form a clot that stops growth. So you want to pay attention to those things. You want to do the same thing for the antibiotics. You need to know, for example, that penicillin targets peptidoglycan, but yet something like tetracycline is targeting the ribosomes in the cell, or that the quinolones target nucleic acids. So pay attention to those things. You could get questions that just ask you straight out what the target of an antibiotic is, or you could get a question that lists a number of antibiotics and asks which one is not like the others. So you might have, for example, 5 antibiotics, 4 of them target the cell wall, but 1 targets a ribosome. You need to know what those antibiotics do so that you can pick out the one that targets the ribosome. So there's that. Know your basic definitions and concepts. What does bactericidal mean? What does disinfectant mean versus antiseptic versus a sanitizer? How is sterilization different from sanitizing? You need to know these basic things. And again, you want to pay attention to what conditions can determine or affect the effectiveness of your antiseptics and disinfectants. One thing that you might overlook that's in the chapter on antibiotics and antimicrobials is how the bacterial cell wall is made. So I went through all the different compounds and proteins involved. We talked about the autolysins, we talked about the transpeptidase, we talked about things like bactoprenol, and how they function in making the cell wall. It's important that you know that process somewhat so that when it comes time to looking at antimicrobials, you know what the target is. You understand how that antimicrobial works. Final thing you want to do is take the practice exam. So there's a practice exam that has questions from the chapter on antiseptics and disinfectants, as well as the chapter on antibiotics. You want to do that a few times because some of the questions on that practice exam are part of the pool for the actual exam. So let's talk about essays now. So there will be 3 essay questions. Here are some hints on things that you want to know while you're handling those questions. The first thing is, remember when I told you needed to pay attention to the cell wall of bacterial cells? Well, one of the questions that's an essay question deals with the cell wall, so particularly the gram-negative cell wall. As you do your review, you want to ask yourself, why is the gram-negative cell wall called toxic? What component in that cell wall is the toxin? And you might further talk a little bit about how it makes people sick. Okay, why does the gram-negative cell wall make people sick? You also want to pay attention to why the gram-negative cell wall makes bacteria harder to kill. All of this is back in that functional characteristics, I believe that's the chapter, functional characteristics of prokaryotes. It's like the 3rd chapter that we did, the 2nd or 3rd chapter that we did this semester. We also talked a bit about the Lau assay. How is that related to gram-negative cell walls? I would pay attention to that. Finally, you should be able to name or list 3 or more antibiotics and disinfectants that are effective specifically against gram-negative cell wall bacteria. Next exam essay that you want to look at has to do with your antimicrobials, specifically antibiotics. What characteristics should all antibiotics have? Now, they don't all have this characteristic, but this is what's desired. You might go back to the quote by Paul Ehrlich that is either in your textbook or in my lecture notes. You also want to go back and look at what broad spectrum means versus narrow spectrum when we're talking about antibiotics. You should have some examples of both types of antibiotic, and then you should be able to discuss the benefits of using these antibiotics, like when would you go for a broad spectrum versus when you would go for a narrow spectrum, okay? And then if there are any downsides to using a broad spectrum versus a narrow spectrum, you should be able to talk about that. So your 3rd essay deals with antibiotic resistance. When you go back and review this topic, some things you might consider include, how is antibiotic resistance promoted? What conditions promote the development of resistance? You might think about ways that people misuse antibiotics, and you should be able to explain how that contributes to resistance. You might talk about how antibiotic resistance is developed. Remember, when we have a population of cells, the cells in that population will have differing resistance to different drugs. So when you add the drug, the most susceptible microbes die first, followed by those that have increasing resistance, right? The most resistant cells are going to be the last ones to die. So how does that promote resistance? You might also want to review horizontal gene transfer. Remember, we talked about that way back during microbial genetics. You might be asked how horizontal gene transfer promotes the spread of antibiotic resistance. Now, you've already answered this question once on exam number 2. But many of you did not answer the question completely, and so you did not get full credit. You want to be sure that you can not just describe the different methods of horizontal gene transfer, but you also want to talk about resistance genes and how they are transmitted by these methods. Okay? Finally, you want to talk about methods of resistance. In your lecture notes, there are 4 ways that bacteria and other microbes can resist antibiotics. They include things like efflux pumps or enzymes that break down the antibiotic, or maybe the microbes have mutations that change the shape of the target for antibiotics. You know, there's 4 different things that are listed. I would go back and review that, because that is something that you need to know for this exam. Now, as I told you, the essay part is going to be closed book. You have 2 hours to complete the exam. I don't expect you to write me the best essay that ever was written. These questions are going to be multi-part. All I really need for you to do is write just a few notes describing the answer to the question. Okay? You only have 2 hours to do the exam. It should only take you 2 hours. So please don't write great essays. Just make notes about the answers to the questions and move on. Okay, just like you don't have a whole lot of time to take the exam, I don't have a whole lot of time to grade them. So help me out. Be succinct with your answers, please. Make sure you read each question well. Over the semester, there have been some people who have not read the question that I've asked, and they've lost points because they never answered what I was asking for. So please make sure you read each question well, and please make sure that you answer what I'm asking and answer completely. If you leave me too short of a phrase, too short of a note, I may not be able to guess, and I probably won't even try to guess what it is you're trying to tell me. So make sure your notes are clear, okay? Your meaning is clear. There are 3 essays. You have to finish each essay in one sitting, but again, you don't have to take all the essays at one time. You can take an essay on 3 different days if you want, okay? Your responses aren't timed, but you do have to finish in one sitting, okay? And again, the essays are closed books, closed notes, open mind. So that's it. I'm running up on 20 minutes. Hopefully that gives you an idea of what to expect on the exam. You can drop me an email and let me know if that was helpful, or if you have any other questions. I'm always available to you. I'll also be available for office hours for the next couple of days. Friday I will not be available, and then Monday through Friday of next week I will not be holding my regular office hours because it's kind of like an exam week. But if you need to see me, you can drop me an email and we can arrange to make an appointment. Okay? All right. Good luck with your studying. I am cheering for you. You've worked hard this semester. Now you get to show me what you've learned, and I am so looking forward to seeing it.